All right, hi everybody. Please ignore my very fancy filming setup, but I thought it would be nice to provide you with another visual on some of the techniques that I'm going to expect of you to use throughout your mystery micro project. You will be working alone throughout the next 2 weeks as you conduct your tests and try to identify the mystery microbe that you have been assigned. You will be expected to do this all on your own without the help of your lab mates and myself. So first, we are going to start with the streak plate. Let's assume that I already sterilized my bench with the ethanol that is found at your table. First, we are going to draw our 3 quadrants, which we have used throughout this term. And then I'm going to turn on my burner. You will be expected to turn on your own burner and use your own burner throughout this project. This is important as I do not want you to depend on others in your class. So I sterilized my loop. I made sure that the entire loop up and down was red hot. This is important so that you do not contaminate your plate. For this project, you are going to be starting with a liquid culture and TSB. You will have a number, and that is all you will have. So first, I'm going to open my tube, and I'm going to run it through the flame very quickly. Notice how I am not putting down the lid onto the bench. It could be a source of contamination, so the best you can, keep the lid and the tube in your hand. I am also going to make sure to work under the area where the flame is, and this is also going to protect your plate from any possible contamination. So I am going to dip my loop into the liquid culture, flame it again, put it down, and I'm going to grab my streak plate. So the first thing you're going to do is use your loop to run across this larger quadrant. This will be quadrant number 1. I like to do a lot of streaks because as you go, you are going to be slowly mechanically diluting the bacteria that you're putting on your plate. After I'm finished with quadrant run, I'm going to flame my loop again. You want to make sure to flame it well. You do not want any residual bacteria on this, otherwise you will not be able to have any isolated colonies. As I wait for this to cool, I'm going to turn my plate, and then I'm going to streak through quadrant number 1. I am not going to take bacteria from my tube. I am done with my tube now. Do 1, maybe 2 streaks, not all the way back through that first quadrant, and then you're going to repeat. I'm going to do my back and forth throughout that quadrant, burn it again. Then I will twist my plate, wait for my loop to cool, and then I'm going to take my loop and run it through quadrant number 2, doing the exact same thing. Maybe 1, 2 passes, and then good spread. And hopefully, when you are done and you have incubated this over the next day or 2, you will have achieved isolated colonies. When I am done, I will flame my loop again, close my lid. Now that I'm done using the burner, I'm going to turn it off just to protect myself. And now I am going to show you how I expect you to label this. This side where I streaked is the side where the agar is. That is where I want you to label. I do not want you to label where the lid is. Otherwise, especially in other tests, if you use multiple bacteria, you will not be able to keep track of the bacteria you're using. In addition, if you were to take your lid off, put it down, you may mix it up with other plates that you have been using. So first, for this project, I want you to write your full name. So I will write Jonna T. Just the initial for the last name is okay. Then I want you to number your bacteria or add the number in. So I'm working with number 13. I will date it. Today is November 3rd. I am using TSA. This is what you will begin with. This is our basic agar that we have been using throughout this term. I would also like you to state the temperature that you are putting this into the incubator. We have the 32-degree incubator, and we also have the 27. It's important to take note that some of the bacteria that will be given throughout this project will do better in one or the other of the incubators. If you would like to do multiple straight plates to increase your chances of getting isolated colonies, you may. I would also recommend putting one plate into the 27 and one into the 32 just in case, because I will not tell you which temperature your bacteria will grow best in. So this is what I expect you to produce. I do not want to see labeling along the side, on the inside, or on the lid. This is how I want it. Along the edges, do not label in the center. Depending on what you will be looking at, this could impede your view. So for the streak plate, this is what I expect.